Positive.
Cool. Morphology.
Positive. The C word.
And then the arrangement is in, yeah, intended clusters. Staphylococcus, I think it's from the Greek, it means like clusters of grapes or something like that. The colony appearance when they grow up, a lot of them are golden, so aureus, in the Latin gold, oro, prefix. Beta hemolysis on blood agar plates, so they do secrete some hemolysins. Let's see, what else? What other types of gram-positive cocci were we introduced to yesterday? Strep, and then there was one other genus. Enterococcus, yeah, exactly. So streps and staphs, we'll talk about, we'll get into steps today, but when we're distinguishing staph from strep, what would be different about the arrangement of this bacteria?
Linear versus clumps.
Streps are going to be in chains versus staph are like clusters.
Typically, yeah, but can we make a definitive identification based off of arrangements? No, just an indication that that's probably what you're dealing with. Good. What about biochemical tests? We talked about two biochemical tests. Catalase and coagulase, good. If I'm trying to just distinguish, let's see, I already have my gram stain done, I've looked at my slide, and I'm seeing that we have gram-positive cocci in clusters, but I want to make sure, so I follow up and do what test first? Catalase, and what would that tell me? If it was catalase positive, it would tell me staph or strep? Staph, good. Catalase negative is probably going to be strep. Okay, so I've got my staph. It could be Staph aureus, or it could be non-Staph aureus. Then what test would I do? Okay, so if it's coag neg, it's a coag neg staph. It's a commons. What were two of those species that we talked about of coag neg staph? We haven't even talked about that in detail yet, but epidermidis was one. Saprophyticus, yeah, good, okay. In terms of some virulence factors, we talked about a bunch of different toxins. We talked about some invasive enzymes that staph produces. Staph aureus, it makes it so virulent. It makes it much more virulent than the other species of staph, epidermidis and saprophyticus. What were some of those invasive enzymes that like virulent Staph aureus strains will produce? Hyaluronidase, which breaks down hyaluronic acid in connective tissue. Good. Anything else? Lipase?
Awesome.
Yep. What does staphylokinase do?
Breaks down clots.
Yeah, it's like fibrinolysin. Yeah, it breaks down those clots. And then coagulase, since we're talking about Staph aureus, which is coag positive, what would that do for virulence? Yeah, it forms that barrier around it, so it's like the opposite action of that staphylokinase. Yep. Okay, good. Now, moving on to talk about some of those toxins. There were a bunch of toxins. The cytolytic toxins. What were some of those, the Greek letters, if you will, for cytolytic toxins? Alpha, beta, gamma, delta. Yep. And then PV leukocidin was another cytolytic toxin that is produced. Not all Staph aureus strains carry these toxins, particularly those strains that carry PBL. Those tend to be associated with some really severe diseases like necrotizing pneumonias, really severe skin and soft tissue infections, things like that. So it's important to remember PBL. It's a big virulence factor, big toxin. We also talked about a couple superantigens that some virulent strains of Staph aureus can produce. What were those superantigens? Some of those enterotoxins, yeah. And then there was another really important one.
Toxic shock syndrome.
TSST, toxic shock syndrome toxin. TSST1. Okay. In terms of the diseases and infections that can be caused by Staph aureus, there were a bunch. What were some of the infections? Okay, endocarditis, very good. Endocarditis. Just in general, hospital-acquired infections could be any number of different things. What about like specific infections that different sites in the body.
Osteomyelitis.
Okay, osteomyelitis, and that is an infection of?
Bone.
Yes, okay, so that's usually a localized infection. If you wanted to diagnose that in the lab, get a lab identification, they would probably have to actually go in and take a little piece of bone fragment and send it off to lab to get a positive ID. What else in terms of like really deep infections besides osteomyelitis?
Endocarditis.
That's Romania? Sorry, that's Romania.
Oh yeah, so what's that? You have bacteremia, what's that? Blood. Yeah, and that, if you have severe bacteria, like lots of bacteria in the blood, that can cause, trigger sepsis, which is immune dysfunction. Cytokine storms. Very good. So I want to go back to the deeper infections. I heard osteomyelitis, which is an important one, but on that same side, there was another one. Arthritis, yes, so it can be responsible for joint infections. And then we can circle back and do those skin and soft tissue infections too. What were some of those skin and soft tissue infections that you can get? Furuncles,
Carbuncles.
impetigo, yeah. Carbuncles, cellulitis, yep, exactly. And then really minor skin infections, besides impetigo is usually pretty minor too. There's folliculitis, yeah, that's very, very, very common. Okay, so we feel pretty good about yesterday. Oh, actually, I missed something. So those were the infections. Toxin-mediated diseases caused by staph, or caused by the toxins that staph produces. So no bacteria required, but most of the pathology is related to the toxins. We talked about 3. Actually, we haven't talked about them. We listed them on a slide, and we're about to. But do you guys remember those diseases?
Toxic shock.
Toxic shock syndrome. Staphylococcus scalded skin syndrome, yes. And there was one more, food poisoning. Yeah, so we're going to talk about those now. Starting with toxic shock syndrome. Okay, so when we were talking back in block 2, we were talking about newer diseases that could be caused by behaviors. And we really didn't have a lot of cases of toxic shock syndrome prior to the 1980s. When a cluster began appearing, like I said, cluster of women, usually in their early years, reproductive years, took a long time, but epidemiologists did trace that back to a source or a behavior or some sort of transmission source, and that was tampon associated. So because of that, for a while, we had most of our association with this syndrome associated with tampon use, menstrual associated toxic shock syndrome. However, the industry did respond. There were like tort lawsuits filed against those companies and a lot of pressure for them to educate the public. So nowadays, this shift has gone where the majority of cases of toxic shock syndrome are caused by non-menstrual infections. We may not always know the point source. Sometimes there are wounds that are seeded, and the conditions are right for strains of Staph aureus that are carrying this toxin, TSST1 toxin, to start producing this toxin and causing toxic shock syndrome. So all that to say is if you see a male who has some of these symptoms that we're going to talk about, it doesn't mean that they can't also have toxic shock syndrome. You shouldn't always associate that with like a 25-year-old woman. Right? Majority these days are actually non-menstrual associated. What does this look like? The bacteria will start as a localized infection. If we're talking about like menstrual infections, this is often in the vaginal tract where you'll have a localized infection. The bacteria have the right conditions, usually related to oxygen levels, to start triggering production of this toxin. And that gets taken into circulation, and then you have systemic effects. Those systemic effects usually manifest early on in presentation with things like this rash that looks like a sunburn. Eventually, that rash, like 1 to 3 weeks later, might be followed by desquamation. What does that sound like? Skin starts sloughing off. Yeah. Even before that, that's usually associated more with convalescence. Before that, this rash is oftentimes associated with other systemic effects that are indicative that you have a super antigen like TSST1 causing uncontrolled immunopathology. So the fever, vomiting, diarrhea, the hypotension, and then in fatal cases, because you absolutely can die from toxic shock syndrome, you have a lot of liver damage potentially, kidney damage. Those toxins can cross the blood-brain barrier and cause neurological problems as well. So when people die, it's usually because of shock and multi-organ failure there. Characteristic for this disease, as I mentioned, was that desquamation. That's usually just a result of the toxicity and your body is sloughing off the outer layer. But what's going on inside is much more harmful. The organ damage is happening. This is a fatal case. You can't see what's going on in the hands and the palms, but it's unusual to have sloughing off in the hands and palms. Palmar and plantar sloughing is characteristic for toxic shock syndrome. Another toxin-mediated disease to be aware of is this foodborne type of illness. It causes gastroenteritis. These are oftentimes associated with food that has been kept at improper temperatures. Temperatures less than 140 degrees Fahrenheit, or they haven't been refrigerated quickly enough. There's like a safe zone where the temperature is too hot for bacteria to live or too cold for them to replicate. Anything in between is considered unsafe long periods of time, like 2 hours, right? So when you have food, particularly things like these protein-rich foods, and that I don't have an explanation. I don't know why staff do particularly well with like egg salads and ham salads and cream pastries. But salty foods too, like ham, that's more intuitive because it is a high salt tolerant organism. Anyway, they start replicating in these food sources at those temperatures, and when they start replicating, if those strains are carrying enterotoxins, like enterotoxin A, B, C, D, etc., they're carrying those toxins, they can produce those toxins and it can get into the food. When you eat that food, those toxins are preformed, which means they're readily activated and they can do damage immediately. And that's responsible for that rapid onset of symptoms. So if you're somebody who's exposed to a contaminated food source and they get sick rapidly, it's probably not due to a replicating active bacterial infection, because that would take longer. It's probably due to a preformed toxin. Okay. You can, this is like I think I mentioned earlier, in terms of exotoxins, this is a little bit on the more heat-resistant side of that scale. So you can heat it up for a little bit, mild reheating, and it can withstand that. If you cook it substantially, you will destroy all of those enterotoxins, but mild reheating, it can survive, those exotoxins can survive that. It also doesn't offer any kind of taste, so you could keep on eating that. Again, this is a dose-dependent manner, so the more you eat this enterotoxin, the more severe the symptoms can be. Let's look at an actual outbreak that happened that illustrates a couple of these points. This was a huge outbreak that happened a couple decades ago in Brazil, in a town, a good-sized town, Minas Gerais. And it was during the ordination ceremony for a priest, so they had food served, and I don't know, they don't have the data in this study. If you're interested, here's the link to that study, or at least the information to find it. So most of this just came right out of the abstract, this write-up that we did. But 8,000 people attended. There was some food source that a lot of those people ate, and 4,000 people got sick from that event. Following the epi from that, so acute gastroenteritis in 4,000 people, about half of those people sought medical treatment. So imagine, this is sort of like a mass hell event almost for even a good-sized town. You have all these people seeking medical treatment. 2,000 people went to the emergency department with gastroenteritis. 26 local hospitals. This is a good-sized town. About 20% of those people were admitted for treatment. They were bad enough where they were admitted. And of those, another 20%, so now we're talking about 80 people or so, were severe enough where they were admitted into the ICU. So those people, certainly we're talking about systemic effects because of this being super antigen and causing sepsis-like conditions in those people. Okay. Of those 81 people, 20% of those 16 people went on to develop irreversible multi-system shock, organ failure, and they died. So 16, we don't really know what the actual denominator is because we don't know how many people consumed the food. But when you have an outbreak of this scale, affecting this many people, there's going to be an outbreak investigation, which they did. This is in the late 90s, so, and in Brazil, so I don't know how they approached it, but they did take a lot of stool from patients. They tested that stool, tried to grow up the usual enteric bacteria like Salmonella and Shigella and Campylobacter. Culture negatives, nothing grew from those. So they went on, with this many people who died, they went on, did very thorough investigation. They tracked down the caterers or the people responsible for the food preparation, and they took specimens from those guys. They took like scrapings from under their fingernails. They took stool specimens. They probably took blood as well, and they I think they swabbed their nares as well for carriage. And they found, at least in a couple of those food handlers, that they were carrying Staph aureus, and then they went on to identify that those Staph aureus isolates they recovered were carrying enterotoxins. I think like 4 of those enterotoxins, A, B, and D, if I'm not mistaken. Okay. So all of this is just to say, like, this can cause widespread, wide-scale problems, or just one-offs. But this was related to food that was kept at improper temperatures and consumed by large amounts of people. So that's a food-borne toxin-mediated illness. The last one is this staphylococcal skeletal skin syndrome. Fortunately, it does have another name. It's more commonly known as Ritter disease, R-I-T-T-E-R. Although this is preferred in the literature, it's more specific. But what this is you get strains of Staphylococcus aureus that have that produce exfoliative toxins, exfoliatins, and these exfoliatins are basically proteases. Proteases do what in general? They degrade protein targets. So specifically, these exfoliated toxins will target desmoglein-1, which is an adhesive molecule that is important in the stratum granulosum of your epidermis. So when they start cleaving indiscriminately, widespread over the body, so this is systemic, this toxin is going all over the body. When it starts doing this widespread, you get disruption of the outer layers of skin, and that manifests as these huge blisters that are called bullae. Bullae is the plural for vulva, but that's what bullae are. They are blisters. And the skin, it can just start sloughing out very dramatically large chunks of skin, large gills. So it's kind of alarming. You can also get this on the palms and on the feet as well. There's a test you can do called the Kolsky sign, where if you just very lightly press against one of those bullae, and if the skin sloughs off, that's considered a positive Nikolsky sign. So it just falls right off like that. And that's, again, because you've had major disruption and damage done to the stratum granulosum. If that patient, usually the patients who recover from this is mostly just affecting the epidermis, and when they do recover, this layer will regenerate, will repair and heal itself. So there are very low mortality rates associated with this, but it does have kind of a dramatic presentation. In terms of seeding, this is oftentimes associated with kids who carry Staph aureus, like they might have impetigo around the mouth and it gets seeded and that toxin starts being produced. Or they have diaper rash and you have compromised skin kind of like in that diaper region or whatever. The Staph aureus get in there and start releasing toxin. Either way, that exfoliated toxin gets released and distributed throughout the body and causes that pathology. All right, so those were the yes, sir.
Can you say bacteria is not covered in the body? Is that like when you take it and try to look at how bacteria cause
Yeah, but that's what I mean. So say you had a mother who brought in her 6-month-old infant and it looked very much like Ritter disease. And you wanted to send specimens off to the lab for them to grow up and see if it's Staph aureus, confirm their diagnosis. if you were to swab the liquid, the serous fluid in this bully, nothing would grow out of it because there's no bacteria there to grow.
Like blood sample.
Possibly, but it's mostly, that could be a negative as well. Like, this is toxin mediated. So if there's a focal point, like the kid has impetigo, the impetigo would have, that would have staph in it. So you could grow staph out of that. If it were diaper rash and you had staph locally there, you could grow out of that. But the bully themselves, this is not bacterial action that's causing the proteolytic cleavage of desmoglein. It's toxin mediated. So from those large swaths of skin that are sloughing off, don't expect to find bacteria. Yeah. And same with like toxic shock syndrome, for example. You're going to be difficult to find the focal point where you'd actually find Staph aureus. And same with foodborne illnesses too, as well. It's hard to find those bacteria. Yes. For the gastroenteritis or? Yeah, that's a good question. So when you have somebody who's presenting with one of these toxin-mediated diseases, I think it's generally pretty hard to diagnose unless there's telltale signs like a bunch of people get sick and you start suspecting a common point source. But if you need to get that confirmed diagnosis, this is oftentimes where, you know, local hospitals, they may be equipped to do like immunoassays would be specific for some of these like specific toxins that are associated with these 3 toxin-mediated diseases. Certainly state labs would have that level of capability where they'd be able to do that, and certainly the CDC. So if it's a big outbreak, the CDC gets involved. Smaller outbreaks, state health departments, state EPI labs will come through. If it's beyond the scope of the hospital, or if the hospital isn't able to, they don't have the capabilities to identify the toxins themselves.
So you're not testing
You might find bacteria depending on what your specimen source is, but with each one of these 3 different examples, like there might be a focal point here that you could get bacteria from, but you might not. So the problem is like what happens if it's culture negative? Can you rule anything out with a toxin disease? It's really hard to rule things out. Same with this, very hard to rule anything out. So it's better if you can find the toxin itself. Better if you can find like the toxin in the food source. You know, you can dissect those cream puffs or whatever. Find toxin in there. Yeah. Oh, sorry. Yeah. Go ahead, John.
Would it
I'll
just
get
be,
you
could you just empirically treat it in? Like if it came in, because if someone was like that bad off, just throw the kitchen sink at it, no,
Yeah, and treatment's a little out of my wheelhouse, but supportive care, if it's going to be something systemic like toxic shock syndrome that could be fatal. I think for toxic shock, since that is so dangerous and life-threatening, there might be immunoglobulins against TSST, possibly for just passive therapy. But supportive care, life support, if it gets bad, yeah. And if there is a focal point, then make sure you have antibiotics to clear up the bacterial infection. And then you had a question?
So I'm guessing even if we took swabs and did a gram scan, we would see anything.
Just depends on what you're sampling and whether or not the bacteria are still there. Yeah, I think it would be very unlikely with the food poisoning example.
Are there any toxoids that we can still compound the actual toxins themselves?
That's a good question. I don't know. I'll have to look into that. I wouldn't be surprised if people had been looking at really highly immunogenic toxins that could be converted to toxoids that could be used to develop vaccines. But I'll look into that. I'm not sure. I mean, the incidence of probably all 3 of these is fairly low, thankfully. But that doesn't mean somebody hasn't tried to make a vaccine, a protective vaccine.
So if these bacteria are producing toxins in the would you not be able to test the blood for the bacteria, or would it kill the bacteria with the toxins?
It kind of depends. If somebody is, so the most serious of those, sorry, I'm going in the wrong direction. This is the most serious, but it's still a localized infection. So if, just to go back to that menstrual associated toxic shock, the bacteria themselves, they are probably localized in one spot. So if you sample that spot, like if you get a good collection of some vaginal specimen and the bacteria are still there, then yes. But in the blood, it depends if they've broken through and like gone through the vaginal membrane and started to get distributed in the blood to become bacteremic and then potentially septicemic. So it's a big if. It depends. Okay. Any other questions on toxin-mediated diseases?
One more question.
Sure.
So at that level when that patient, that picture was at that time if we administer antibiotics, would it not be effective? Is it gone already out of our hands?
Well, this is an autopsy photo. She's already gone, but I don't know when you'd have to administer antibiotics. You'd probably be very specific to that case. Yeah.
I got a question.
Yes, sir.
For this, for the food poisoning case study, are we gonna have to know that slide in terms of like the relevance of like the food poison, or we gonna have to know like, or is this like an FYI slide?
It's not an FYI. Yeah, there's information to be distilled from that. Any other questions? Okay. So, mostly for those cases where we have actual bacterial infections, like the skin and soft tissue infections, osteomyelitis, the pneumonia, when we have something bacterial to culture, this is how we might go about it in the lab. There are lots of options. Staph aureus is not really a fastidious bacteria. It's very cooperative. It's easy to grow. So we get any number of specimen types, depending on what the infection is down in the lab. We usually grow it on blood agar, but because it's so common, we also have a lot of different selective and differential media types that we can use as well, including that mannitol salt agar, which is good because it is so salt tolerant. Depending on the strain and what kind of hemolysins or cytotoxins that it produces, it might have variable hemolysin patterns, but beta is very common, beta hemolysis, like complete hemolysis, with highly virulent strains of Staph aureus. Microscopy we've talked about before. Some of those Staph aureus isolates or strains will produce capsules, some of them don't, but usually the more virulent ones do. We talked about the biochemical identification, catalase positive, coagulase positive. But latex agglutination tests we haven't talked about a whole lot. We talked about agglutination as a principal last block. But with these particular targets, these are two very popular targets if you want to use agglutination to see if you have Staph aureus. Protein A, as you know, coats the exterior of those bacteria. So very immunogenic, very easy to pick up in lab. So you can see if it has agglutination positive reaction there. Or if you have a more specific question, like you want to know if that bacteria is a methicillin-resistant Staph aureus isolate, you can look specifically for PDP2A. You guys remember what PDP2A is?
It's a binding protein. Let's do it.
Yeah, but what's 2A? Yeah, that's a form that the bacteria will make when they mutate and have a different type of PDP. Functions the same way, but it confers resistance to penicillin. So, and the beta-lactams that bind to PDP. So if you want to answer that question, looking for presence or absence of PDP2A specifically with a latex agglutination test would be that answer. As with some nucleic acid-based tests, you can look specifically for whether or not that isolate is carrying the methicillin resistance gene. It's called the MEC-A gene. There's also one called MEC-C. Yes, sir.
What is it about the latex that makes
Yeah, so latex is just like a substrate that you can conjugate your reagents to, if that makes sense. Those commercially available reagents that we use for immunoassays, they're oftentimes conjugated to latex beads. It's just a good substrate for that. There are others as well, but okay, so those screening tests, the cartridge on the bottom is showing you what a little cartridge looks like for the gene expert, but this is a very rapid PCR screening test. It just tells you it's looking for the MECA gene, takes about an hour, and it tells you whether or not somebody is potentially colonized with MRSA. So you can swab their nares, stick it in that thing, pop it in the machine, and get your PCR results. Very quick. MALDI-TOF, we talked about briefly. That's that mass spec instrument, super nice, super fast, very expensive, but increasingly we're using that at DoD clinical labs. A lot of our labs now have MALDI-TOFs. And because there is a lot of antibiotic resistance, these students do not really have predictable resistance patterns. You're going to want to do AST testing when it's a serious infection. Somebody has a little impetigo, whatever, folliculitis, fine. I'm talking about like really serious staph infections. You need to do AST testing. A couple slides talking about resistance because that's really important with Staph aureus and the history of resistance to Staph aureus. So this bacteria used to be pretty susceptible to everything. When did penicillin come on the scene about? Yeah, like by World War II, it was in clinical use. Penicillin resistance developed quickly in Staph aureus. They picked up penicillin resistance very quickly. They were able to cleave. They were able to produce beta-lactamases and start cleaving those beta-lactam rings in penicillin. So by the late 50s, most Staph aureus isolates were already resistant to penicillins, particularly penicillin G, which is used for what? Anybody know? Versus like, you don't have to know for this course, but like, is it IV or is it oral? IV, that's right, yeah. So penicillin V is oral, penicillin G is IV, so this was for several decades used to treat really severe systemic infections. And quickly, when we had resistant isolates start to develop, that was no longer an option. So then we switched to semi-synthetic beta-lactams like methicillin and oxacillin, which were more stable to beta-lactamase cleavage or inactivation. We started introducing methicillin around 1959. Within 2 years, by 1961, we had pretty widespread or the development of methicillin-resistant Staph aureus isolates. So over the next 2 decades, that methicillin resistance, it was selected for, it was amplified, and became like a global thing. So now we have a lot of widespread methicillin resistance in Staph aureus strains. So that was a heavy blow because methicillin was a good antibiotic to use. But yeah, it just kind of illustrates how quickly you can get widespread resistance. There's no such thing as PRRSA because penicillin resistance has been around for so long that it's just sort of associated with, like, you wouldn't even think to treat Staph aureus with penicillin. Okay, this last note on community-associated MRSA, it is worth having this conversation a little bit about community-associated MRSA versus hospital-associated MRSA. When we first started seeing resistance to methicillin, where do you think it appeared first, in the hospital or out in the communities? In the hospitals, where all the sick people are, all the people who are inpatient populations. So these MRSA strains that started developing in the hospitals eventually started trickling out into the community, and we had increase of community-associated MRSA outbreak illnesses as well. But the patient population, when you're talking about community spread, we're talking about just your average person. There's no predisposing conditions, no comorbidities. It could be anyone in this room. In fact, military people are one of those populations that are likely to get community-associated MRSA, and that's because of close contact. If you have heard of MRSA before and some of the activities that are associated with community-associated MRSA, what are some of the ones that pop into your mind? Wrestling. Yeah, there's been some really big, notable outbreaks and transmission of MRSA among wrestlers because super close contact. That holds for people who are working out in the gyms and they're laying down on the same surfaces, using the same equipment. So team sports, just close contact in general, MRSA transmission out in the community. in hospitals, we know that Staph aureus has a pretty robust, thick peptidoglycan layer, right? So it can be transmitted easily on fomites, and that's concerning for hospitals. That's a common way of transmission in hospitals. That's why infection control teams work so hard to decontaminate surfaces. In terms of medical history, again, there's nothing really wrong with patients, typical patients who community-acquired MRSA, they just are exposed to it. With hospital-acquired MRSA, these people oftentimes have predisposing conditions. They might have had recent antibiotic use. They might have just recovered from an upper respiratory infection, and the respiratory tract is beaten up, extra susceptible. In terms of virulence, the cytolytic toxin I was mentioning, the Panton, Valentine, leukocidin, we don't see that as often, but we see that more often in community-associated transmission in those strains. And then finally, with antibiotic susceptibility, because you're in a hospital, there's more transfer of antibiotic resistance genes in the hospital, so you have more drug resistance, typically with those isolates than you do out in the community. We've talked about this mechanism before, so hopefully this is pretty accessible. But this cartoon shows very clearly how you go from being susceptible to penicillin on the left there. Penicillin binding protein is being bound selectively by methicillin, for example, and it's causing it to disrupt peptidoglycan crosslinks. Versus MRSA strains that have that altered form of PBP, PBP2A, which the methicillin is no longer able to bind to, and because of that, you maintain the integrity of the cell walls. The peptidoglycan layer stays intact. Okay, so as we started to get increasing incidence of MRSA, providers had to start relying on other antibiotics increasingly. We haven't talked about these other antibiotics much. Ceftalaridine, that is a notable cephalosporin. It actually was developed to bind to PBP2A specifically. So we're seeing some resistance already to that. Trimethoprim, sulfamethoxazole, that's the Bactrim, more commonly known as Bactrim. You guys know what that targets, right? Folic acid synthesis pathway, exactly. Linolazid, this targets protein synthesis, and this is a membrane targeting, it's a fairly new class of antibiotics. But vancomycin in particular, this is what I want to talk about. Vancomycin was popular because it was used to very successfully treat bad, serious, systemic Staph aureus infections. It has good activity against furin positives like Staph aureus. So it was used, and unfortunately we started seeing resistance patterns starting to develop in the 1990s, and by 2002 we saw resistance in the United States. There were several layers of resistance. The first that we started to see was called VISA, and then later we started to see VRSA, so we'll talk about the difference between those in a second there. First, the mechanism of action for vancomycin. We know that this antibiotic it actions on cell wall synthesis, but it's not the same as a beta-lactam, right? It's in a different class. So the mechanism is different in that, well, first, normally what happens, you have your NAGNAM and your pentapeptides, right? Then you have your your PBP, which is a transpeptidase, and that action is just cross-linking those pentapeptides in that peptidoglycan layer. What happens when you have vancomycin is that you literally have vancomycin binding to that last amino acid on this pentapeptide, and it blocks it. It blocks the transglycosylases and the transpeptidases from doing their thing and facilitating that cross-linking. So the action is steric hindrance. This is a physical block. Vancomycin sits there like a cap, and it prevents cross-linking. Those two levels of resistance I mentioned, the first two appear with this VISA intermediate level, and that was simply when Staph aureus isolates started to increase the thickness of their peptidoglycan layers. The thicker they got, the harder it was for that big vancomycin molecule to diffuse through that layer and start interfering with peptidoglycan synthesis. Vancomycin, by the way, is a very large antibiotic. So the thicker this layer is, it slows down the progress of vancomycin moving through, and it traps it into those peptidoglycan layers. But that doesn't stop it, and you can compensate by administering heavier doses of vancomycin. But as you know, that's a bad thing, right? You don't want to keep administering heavier and heavier doses. So this is concerning just on the merits, the fact that we have VISA isolates. But then we also, unfortunately, started to see these VERSA isolates. This is still relatively uncommon. When we do identify VERSA isolates, it's cause for alarm. It's a very, very, very bad thing to see. But we get high level resistance, which means no matter how much vancomycin you give that patient, it's just no longer going to work because the target has been completely nullified. Going back to our normal diagram, what happens is you can no longer have vancomycin binding to the end of that pentapeptide cross-link at that point. And that is because that the last two residues, you have a diala-diala normally, are replaced by a delactate-diala motif. Okay? And because of that, vancomycin doesn't bind at all. Putting that all together, I know this diagram looks kind of scary and horrifying, but it puts together some concepts that we've talked about before. Starting with the source of this resistance operon here, this is the VAN8 operon that encodes for the enzymes that alter that pentapeptide structure and make it so that vancomycin can no longer bind. So this comes from this genus that we mentioned yesterday, Enterococcus. Enterococcus carries a plasmid with VAN8 operon on it. We talked about genetic transfer mechanisms before. What were those three mechanisms in between bacteria? Transformation, transduction, and conjugation, right? So typically through either conjugation or transformation, an Enterococcus bacteria that is carrying those plasmids with VAN8 gene on them will transfer those genes onto a susceptible Staph aureus isolate. This is called VSSA. So it's totally susceptible. It acquires these plasmids and then it becomes resistant itself. Those Enterococcus, they have had vancomycin resistance for a while. So we have VRE, vancomycin resistant Enterococcus. That's a problem in and of itself, but that's how it's transmitted. Looking at the plasmid itself and the operon that's expressed on this particular plasmid, you have sensory regulatory elements upstream, and then downstream, you don't need to know what these do, I just want you to know the principle here. You have some genes in this operon that encode for different enzymes, and basically when those enzymes get expressed, like in the presence of vancomycin, that's the inducer, when they get expressed, the job of those enzymes is to kick off that diala residue and replace it with delactate. Vancomycin can no longer bind that. That's all we need to know. Okay. But it brings together a couple different concepts that we've talked about so far. Yes, no?
Do you have testing to identify VERSA versus
If you could look for the VAN8 gene with PCR, and if you detect VAN8, it could be VERSA, and if it has the right phenotype, as in it's showing resistance when you're doing AST testing, yeah, I think that would be a pretty solid way because that VAN8 operon probably wouldn't be on those VISA isolates. Yeah. Any other questions about this deck? Oh, sorry. Yeah.
Have they made a type of vancomycin or they in the process of finding that binds to DLAC?
That's a good question. Maybe, but if they, yeah, I mean, I don't know if it's worth exploring because if they did invest millions into developing another drug that would bind specifically to delactate, those bacteria would be under selective pressure and we'd have the same problem. And we've learned through the last 6 or 7 decades of antibiotic overuse and misuse that this happens. And if it happens in one type of bacteria, like Enterococcus, it is entirely possible to have transmission of those resistance genes to other bacteria as well. It's a very frustrating issue that we've learned painful lessons from.
Just to clarify, man, it's still susceptible to vancomycin just at a higher dosage.
It can be. It's kind of in that definition intermediate range. It would be something you'd want to do AST testing for and see where it falls, like that specific isolate that you think is this. How is it behaving when you do like an AST test with it? Was there a question over here? No. Okay. All right. I've got 13:48. Let's take a 10-minute break, and then we'll start on the Streptococcus deck. We won't go very far to the allergy as out soon, but I just want to get through like the intro material so we can finish that deck tomorrow. So 13:58. Let's meet back and we can start again.
So we just don't do it.
Are we not worried about the last 4 slides?
Oh. Yes, I'll pick up on those when we come back to the break. Thank you so much, John. I appreciate that.
We have homework too.
So steady.
No, but if you read the last, I feel like that shit.
I didn't see that right at the top, and I was like,
Yeah, I was even looking
Full belly. I'm just like, saving lives. Like,
saving grace.
Yeah.
Saving grace.
Wait, was that during this time? Yeah? Probably not.
Really? No, his thinking secret. His thinking secret. At least me. That's probably how I got. It's been helping. I mean, they're not like tigers. I mean, I almost got it. I almost got it. There's not one question. Now one question. Well, he's been keeping it to himself. You're not even so different.
There's 2 slides of notes.
Yeah, I think in some ways they
Fine.
but in some ways
That was already on the list.
explain to me like track.
Where did you get that? Nobody ever looked at it. You're right, you're way.
I mean, as long as you isolate the patient. And then you incinerate the room.
Well, it's not.
This test is gonna be rough. Good thing is, this weekend we can touch it. We can afford it. My dad's coming down that weekend. Tell him to entertain himself. I'm probably gonna have to come over like 2 of those days. I'm gonna have to go this so many times. I'm gonna have to do a lot of Donovan was saying that they all look the same. Same, sound the same, do the same stuff. Like questions asked. And I'm just trying to make sure that I'm not looking at the videos that are in there. Is the PI tomorrow? Next Wednesday? And these 2 days we don't have after lunch. We don't have after lunch? No, Jake's gonna publish it, but I think he did. So biochemistry gets up here. So many days. how did you do the review here? We were saying we wanted her to do a review here because then we studied because we were reading how it actually hurt. So we were saying like study as much as you can and she just goes over a review. So we know going into it. Because that review hurt me. I like that review. I thought it was good. That hurt because she said where to dig into. You just didn't dig. I went back to all the slides. I said you didn't. I said you only studied Sunday night and Monday morning when I was looking through shit, like just to like refresh it.
That's just like the location, like what city I just don't listen to the whole thing. I'm not how many patients in a hospital are in high school?
No, she's actually gonna ask you what the DOI name is. Things are published. She said she wants us to know this stuff. What's on the case? Come on. Let's not test it.
If you don't know this, you're bad behind.
Test this on a study case. Which, yes, is relevant. But
It's never a complaint. You just fill it out verbatim. His feet still on his That's like you want to play game. All the patients, like the numbers of this rabbit. She's like, hey, there's a lot of questions on this. It's only one question.
Let's knock out those remaining 3 slides. Thank you again, Sean. No, I do apologize. They're important, but not nearly as important or as intensive as staff RA. So we can go through them fairly quickly. In terms of the cons, the colloid negative staph species, there are more than just epidermidis and Staphylococcus. There's Lachmanensis and Hemolyticus and a bunch of other species. But they are usually associated with less, they don't have as much virulence, and so we tend to question them when they do show up on clinical specimens. I'll talk about that a little bit more. But the two to kind of put on your radar above the others would be epidermidis and Staphylococcus. And are associated with different things as well. And so the first one I want to talk about is epidermidis. It is probably, its unique feature is it probably is the most abundant bacterium on the skin. Virtually everybody's colonized with it. It has a little bit of a mutualistic relationship with us in that it provides protection on the skin. It helps colonize us and keeps that niche occupied so worse bacteria don't occupy it. So generally, not a problem at all. When we recover it in the lab, oftentimes it is a contaminant. So somebody has like a infection and you submit a blood specimen down to the lab for culture, and we identify epidermidis in that culture. We view it with skepticism initially. There's some criteria for it to meet in order for us to be convinced that it's actually causing infection in that person. Right? But the reason why is because since it is colonized in your skin, if you don't disinfect that skin area, the epidermidis is local on the skin. It's going to get into that blood specimen. So when you submit the blood culture, what's going to grow is going to be that non-problematic commensal that is Staphylococcus epidermidis. Okay. It can be a problem sometimes though. So if you submit multiple blood cultures and they are positive for epidermidis, so you've taken a blood culture from one site and taken it from another site and it's continually coming up positive for staph epidermidis, that might be your smoking gun that it is in fact epidermidis. But like I said, to be interpreted with caution. In terms of virulence factors, you don't have nearly as many virulence factors. Its main thing is the fact that it can attach to things really well. So it's good at forming biofilms, and for that reason, it's associated with some of these like catheter-associated infections, heart valve. It can grow and help heart valves, prosthetic joints, things like that. If it is identified as a problem, another secondary issue is that you do have some antibiotic resistance associated with epidermidis, so you do need to do AST testing. But by and large, that's kind of run down for staph epidermidis. Yeah.
So would this kind of be like whenever you're taking a blood culture, you'll like wipe off their site with like chloric acid instead of alcohol? Is that because alcohol might not kill stuff that could contaminate the specimen?
I don't know exactly. I've never done phlebotomy work or taken blood specimens myself, but like they do have very specific protocols to avoid this exact thing. And on the lab side, I can tell you from having worked in the lab, we get a number of contaminants. So even with those protocols, it's not unusual for the lab to grow up staph epidermidis. And then it looks sketchy. It looks suspicious when it grows up in the lab. It's like you only find it from one blood bottle, growing in one and not the other. So those are the types of things where it tip us off to it being a contaminant rather than like a real etiology. This biofilm, this electron micrograph shows the biofilm capabilities of staph epidermidis. You can see it growing on the substrate, going up in layers, and binding very efficiently because of those adhesins that it expresses. The other one to be aware of, this is actually pretty important, Staph saprophyticus. It is a major cause of UTIs, real deal. UTIs carry their own trickiness when you're working them up in the lab because when you have urine flow, you do get some bacteria just from exiting the body. So it depends on the urine source. Is it clean catch? Is it pubic urine? But for the most part, if you see lots of Staph saprophyticus growing up from a urine specimen, especially in this one key demographic, which is women of childbearing age, it could be the cause of their UTIs. That too is linked to their ability to form biofilms so they can adhere to the epithelial cells in the urinary tract very efficiently. But beyond that, you don't usually see a lot of severe infections unless the patient themselves has some sort of comorbidities. In those cases, the complications are listed on the slide where you get more serious infections of some of these genital urinary structures. Of those, the urethritis, epididymitis, and prostatitis. The acute pyelonephritis is the most concerning of those. It's the most severe one. This too is a commensal. It is found normally in the vaginal tract, so it's just normal flora. E. coli causes about 70 to 75% of UTIs, but this is up there. It causes anywhere between 5 to 20% of UTIs. And you can see this very quickly when we grow up a plate overnight from the urine culture. It's a gram-negative rod, then it's E. coli, it's gram-positive cocci. It's more than likely going to be Staph aureus. And that is it for the staff deck. Any questions on this? Okay, I'm just going to switch over really quick and pull up the strep deck. One second. Okay, so just need to get about a few slides deep into this so we can finish it tomorrow. With this genus that we were introduced to yesterday, we have more clinically important species to cover on this, unfortunately. And towards the end of the deck, we'll talk about some of the more clinically relevant Enterococcus species as well. So 2 different genera in this one deck, but the vast majority of this deck is concerning Streptococci. Like with Staph, gram-positive cocci, but again, those arrangements are going to be in sheet chains. So here we can see some gram stains of different types of strep species. We have Pyogenes, which is probably the most virulent species for this genus. Nice long chains. It's staining rather darkly here, but yeah, gram-positive cocci have long chains. For a gram stain, or sorry, for culture. And then from a blood specimen, you can see a different species called Strep mitis growing again, very nice long chains. With the scanning EM, you can see the reason why you get chains instead of clusters. You have division, just planar division. And these cells divide by binary division, unlike Staph, where you have that ability to divide in multiple planes. Strep pneumo, Streptococcus pneumoniae, is another really important member. We'll talk about that in more depth, but that would be the exception for this arrangement. You don't usually see those in long chains. You see them oftentimes in pairs. So pairs of strep pneumo. Catalase negative, we've talked about that before, and that is sort of related to the fact that they are aerotolerant anaerobes. It's a little bit different than facultative anaerobes. They merely tolerate oxygen conditions. They don't do as well as Staph do. So that's the reason why they're catalase negative. They don't thrive in those oxygenated environments, but they can survive in this. And because of that, the types of infections that they can grow in are diverse enough. You can get them in wounds that have not a lot of oxygen in those environments. You can get them in deep tissue infections like necrotizing fasciitis and other really, really deep infections because of that aerotolerance. Like Staph, they are also normal colonizers of the human skin and the upper respiratory tract as well. We have a lot of different species that we carry with us that are commensals. Not all of them are going to be pathogenic. And even the ones that are highly virulent, like Strep pyogenes, we can be colonized with without any issues. It's just something it's better not to be colonized with them because it's a risk factor. The genus does have quite a few species, and because of that, over 100 different species in this genus, we've developed some really complicated, intricate classification methods historically when we didn't have molecular testing for them. So for example, the Lancefield groupings, this is probably over 100 years old. It is a very old testing system that's been done since the 1930s or 1920s. And it's based on the presence of an antigen in the cell wall of these gram-positive cocci. It is not present in other genera, so it's not present in staph. That antigen is called C substance, and when you do immunoassays looking for particular C substances, those commercially available antibodies will bind to one of these serogroups. There's A through U, alphabetically, they're lettered like that. There are some gaps. The only ones that are really important to know are groups A, B, C, and D. So those are the ones we'll primarily talk about, and of those, group A and group B are really the important ones. Not all strep species are typable. Some of them don't have Lancefield groupings because they don't have a specific antigen in that C substance in the cell wall. So we'll talk about those as well. So there's the Lancefield grouping, and then there's also the hemolytic pattern on the plate that gives us an early indication about what strep species we're dealing with. Beta hemolytic species like Strep pyogenes are considered to be the more virulent ones. So you have Strep pyogenes, and then you have Strep agalactiae, group B strep. Those are major pathogens. Strep pneumo can also kills a lot of people annually. Every year, a lot of people get these bacterial infections, particularly in the lungs with strep pneumo. That causes alpha hemolysis patterns on a plate, typically. And then you have some other genuine pathogens that cause no hemolysis at all. So gamma hemolysis. Both of these different classifications are used in conjunction to make preliminary identifications of streptococcus species. So for example, with Strep pyogenes, that is a group A beta hemolytic strep. We use them in conjunction still today in the year 2025. We call them that. So all of this holds true today, and you need to know it as providers because this is how the lab reports these results out. So it needs to mean something to you when you get that report back that you're doing group A strep or group B strep. When you're doing this Lancefield test, literally you take a colony from your plate. This would be your unknown. So unknown antigen, commercially available antibody. You smear a little suspension of that unknown bacterial colony onto one of these little wells on this card, and then you add this reagent. That reagent just has an antibody, and then you mix it together. If it glutenates, it's a visible clumping reaction. Hopefully this is sounding familiar. Then you have a positive group A or group B or group C. But that's determined by which one of these antibody reagents you're adding to that picture. More on classification scheme here. So on this particular flow chart that we're looking at, it's starting from when you were looking at the gram stain on a microscope. And this is the first thing you see. So you see gram-positive cocci in chains, and you're automatically thinking, this is probably strep, right? So the next thing you could do is a catalase test. It would probably be negative, so more certain that you're dealing with streptococcus. Then you can go back to your plate and look at the hemolysis patterns of those colonies. And that is when you're really going to start getting a good indication of what species you're looking at. If you have a beta hemolytic colony, what species, according to that flow chart, or what two species could you be looking at potentially? Pyogenes or agalactiae, exactly. And then you have another test called bacitracin sensitivity test. You don't need to know that, but that can help very quickly distinguish whether it's Pyogenes or agalactiae. Other hemolysis patterns, the alpha hemolysis species would be strep pneumo or viridin strep, which we'll talk more about. And then the ones that have no hemolysis, again, just because they aren't producing hemolysis doesn't mean they're not serious pathogens. So we have Streptococcus bovis and then Enterococcus. There's a couple species there. A few more notes. I had mentioned that not all Streptococcus species have that C substance in their cell wall, right? So if you see something that says none, like the viridins group, there's no Lancefield group associated with the viridins group because they don't have that C substance. So that's what that means. And same for Strep pneumo, unfortunately. It doesn't belong to a Lancefield group. Also, there's sometimes a little bit of confusion when we're talking about viridin strep. Some people reverse the order and call it Strep viridans, and it starts sounding an awful lot like a species, but it's not. It's a group of species that have similar characteristics. It's the viridans streptococci. So here we have 5 different species that belong to this group that all have similar characteristics, but there are nuanced differences, and none of them have a Lancefield grouping, and most of them are going to be alpha-hetolytic. And then lastly, most glaringly, is the fact that Enterococcus is showing up on this list of streptococci. And that is because this different genus has C substance in its cell wall. Because of that, it types as group D strep. Also, for historical reasons, Enterococcus and Streptococcus used to be in the same genus. So just, I guess, for historical apathy, we kind of group them together still, but they are 2 separate genera. Unfortunately, they do behave similarly. They cause similar diseases. They can cause UTIs. They can cause wound infections together. They can cause respiratory infections. So it makes sense to keep Enterococcus and Streptococcus together and refer to them as group D, but they are separate genera. And the chemolytic patterns here note that they could be either partial hemolysis or they can be no hemolysis. It just depends. All right, here we see some nice streps out on a gram stain. Very, very commonly encounter bacterial pathogens. We have them on our skin. We have them heavily in our nasopharyngeal cavity. So as opposed to Staph aureus, they can occupy a similar niche, but Staph aureus like to be where in the nose? In the anterior portion of the nose. These tend to be farther back, deeper into the nasopharyngeal cavity, but they can kind of be in the same areas, and they do compete with each other. They produce enzymes that are sort of meant to destroy one another because they're competing for nutrients and competing for resources in your nose. Unlike staph, they do not survive well out in the environment, and that has implications for how they're transmitted from person to person. It requires more direct contact, typically, to transmit Streptococcus species than it does for staph species. With staph, remember, you can have fomite transmission because they are resistant to drying. With strep, it's more like direct person-to-person transfer if I'm colonizing my nose and I'm sneezing or I'm talking to you and releasing or shedding strep species potentially. But it's not going to be one of those things where you pick up strep necessarily because you touched a doorknob someone else touched yesterday. In terms of pathogenesis, we have a couple of major diseases that we'll talk about. Depends on the type of species, but this is an introduction. Some of the important ones are pharyngitis, which is what? Sore throat, yeah. Cellulitis we talked about, that's a deeper soft tissue infection, deeper layers of the skin. And then necrotizing fasciitis, mentioned a little bit, but we'll talk more about that. There are a bunch of virulence factors when we're talking about Streptococcus pyogenes, this really virulent species. So we'll talk about those more on the next slide. But in general, you do not necessarily need a lot of this organism to cause disease. They're low ID50. And another concern is even after you have those infections and you've cleared them, you can have some really significant post-infection sequelae that we'll talk more about. Okay, I think we should call it before we get into this slide, if you guys are amenable. We'll pick up tomorrow talking about pathogenesis. I think we should be able to get through this stuff tomorrow, but if not, that's fine. We'll do it next week. Let me know if you have any questions, and I'll see you guys tomorrow at